Hello, my name is Jordan Kushner, and today I will provide a lesson covering some of the topics that are discussed in Chapter 55, Hemostasis and Thrombosis. Here is my name and date. So, before we dive in, here's a list of lesson objectives covered throughout my lesson. By the end of the lecture, students should have a good understanding of why hemostasis is important in achieving health and how it aids in the understanding of various disease processes. Students will be able to illustrate the important biochemical processes that occur from platelet activation and the multiple steps involved with platelet plug formation. Lastly, I will go over one of the more common drugs, aspirin. Students will be able to understand how its mechanism of action inhibits platelet aggregation. Our body has unique properties that allow blood to move freely and transport important chemicals and nutrients to all our body tissues. This is a crucial aspect that is vital for life by allowing our tissues to remain healthy so they can carry out their intended purposes. While it is important for our blood to move freely throughout the body, it is equally important for our bodies to be able to stop bleeding if an injury to a blood vessel does occur. The concept of coagulability relies on various biochemical processes that are vital for survival. If our blood was not able to form a clot, we would be at a high risk of death due to blood loss just from minor injuries such as a paper cut. Some of the leading causes of death result from issues within this concept. When our bodies form clots when they shouldn't, their effects could be devastating, such as myocardial infarctions or strokes. Many of the drugs used to treat these thrombotic diseases are designed by their ability to manipulate the biochemical pathways involved in either prevent clotting or encourage it. An example that I will touch on later in this lesson is aspirin, which is a common platelet inhibitor. We already talked about why our bodies need to be able to control and minimize bleeding when there is an injury that causes blood loss. Now I will get into the biochemical processes involved in one of the steps that helps us achieve hemostasis, platelet plug formation. When there is an injury to the vessel wall, the endothelial cells secrete an important protein called von Willebrand factor. This enables our body to start the process of forming a platelet plug in order to seal off the injured vessel and minimize and stop blood loss. When the von Willebrand factor is secreted or released, it causes platelets to move toward it. As the platelets move closer, they will bind von Willebrand factor, and this binding occurs via their glycoprotein 1b receptor on the platelet. Once this binding occurs, it activates the bound platelets to start secreting chemicals, including ADP and thromboxane A2. When these chemicals are released, they bind to specific G-coupled proteins on the platelets and cause them to migrate to the site of injury where the platelets are already bound to the von Willebrand factor. This process can be referred to as platelet aggregation. As more platelets start aggregating to the site of injury, they start binding to each other, forming a nice platelet plug to minimize bleeding. So we talked about how platelets bind to von Willebrand factor through the glycoprotein 1b receptor. Where they bind each other is a different receptor. This receptor is the glycoprotein receptor 2b3a. Fibrinogen, which is also known as factor 1, is what links the platelets together and allows this binding of the glycoprotein 2b3a receptor to occur. And this is our platelet plug. This is an image that is in our textbook that describes the various processes involved for platelet activation and plug formation. There's a lot going on during this process that I have not delved into specifically in this lesson. I included this image to provide an illustration of how platelets bind each other via the glycoprotein 2B3A receptor. This is best seen on the bottom of the image labeled B. You can see glycoprotein 2B and 3A has a resting state versus its activated state. Fibrinogen or factor 1 in our blood causes the platelets to bind each other when this receptor is in its activated state to form the platelet plug. So you could see in the resting state, fibrinogen is just hanging out, and then in the activated state, it helps the platelets bind each other. This is an image I like better than the one that was in our textbook that illustrates the biochemical process of platelet activation and plug formation. The link is provided under the image if you would like to review the article, which is on Science Direct. The article talks about how Staphylococcus suberectus inhibits platelet aggregation. So some of the things on this image are not useful for what I'm trying to explain in this lecture. The image does a good job of illustrating parts of the platelet plug formation that I did cover. Look at the top left corner. This illustrates a damaged vessel wall. You can see von Willebrand factor labeled VWF in the top left corner that binds to the platelet via the glycoprotein 1B receptor. This activates the platelet to secrete various chemicals. We talked about ADP and thromboxane A2, which is labeled TXA2. So the release of these chemicals further stimulates the process, and platelets start moving to that damage on the vessel wall. And then platelets begin to start binding to each other. This binding occurs on the glycoprotein 2B3A receptor, and you can see they are linked together with the help of factor 1, which is fibrinogen, during the aggregation. I'm sure we all heard about the drug aspirin before, as it is a common over-the-counter pharmaceutical. You may know people who use this drug that have previously suffered from a myocardial infarction or heart attack. So this disease results when a blood clot occurs in the coronary artery, preventing blood flow to the heart muscle. Generally, a cardiologist can place a stent, which frees the blood flow through that artery. Aspirin is then prescribed to these patients to prevent a blood clot from forming within the stent that has been placed in the coronary artery, ensuring the heart muscle can maintain perfusion and get all the blood it needs. The mechanism of action that allows aspirin to do its job is by inhibiting platelets, specifically by inhibiting or stopping the platelets to secrete thromboxane A2, which we have learned earlier is an important chemical that aids in platelet aggregation and plug formation. It is important to understand the potential side effects that come along with aspirin use. Because we are inhibiting part of the process for blood coagulation, these people are at increased risk for bleeding. Aspirin is used to help prevent several thrombotic diseases and is considered a blood thinner. So here's an illustration that shows how aspirin works. Aspirin causes acetylation, where the acetyl group blocks the catalytic site on the platelet, thus preventing chemical release of thromboxane A2 that is used to stimulate platelet aggregation. I found this image in my research article, in a research article that talks about the clinical utility of using aspirin specifically in cardiovascular disease. So now we will get into some of our questions. So why is hemostasis an important concept in maintaining health? The correct answer here would be answer A. It controls and stops bleeding from blood vessel injury. Question 2, what receptor on the platelets binds to von Willebrand factor to initiate the start of platelet plug formation? And the right answer here is going to be choice B. It's the glycoprotein 1B receptor. So the glycoprotein 2B3A receptor is where platelets bind to each other, not von Willebrand factor with the help of fibrinogen. And thromboxane and ADP are the various chemicals that assist with platelet aggregation. Question 3, during the start of platelet plug formation after platelets bind to von Willebrand factor, what chemicals are released that allow further platelet aggregation? So the right answer here would be choice D, thromboxane and ADP. Fibrinogen, or factor 1, is a protein in the blood that helps platelets bind each other during plug formation. So that's not the right answer. Question 4, what helps bind platelets to each other on receptor glycoprotein 2B3A? And the right answer here is going to be choice A, fibrinogen, or factor 1. The other answers are important proteins, but they do not specifically allow platelets to bind each other during plug formation. And then our last question of the day is going to be glycoprotein 1B receptor binds to what glycoprotein for the start of platelet plug formation during hemostasis? And the right answer here, hopefully you guys know this one, is answer A. So that's going to be von Willebrand factor. Plasmin, fibrin, or heparin sulfate do not bind glycoprotein 1B during plug formation, as talked about in my lecture. And here's a list of all the reference I used when creating this lesson, and hopefully you guys learned a little bit about hemostasis and thrombosis. Thank you.